Hello and welcome to another video for our flipped classroom for bio. Today we are looking at an interesting dot point. It looks kind of big, but hopefully we'll get it down into little manageable, digestible pieces. It's all about how stuff is carried in the blood. So let's unpack our dot point. We've got our first verb here, which is identify, our first and only verb, which is identify. And hopefully you guys are getting used to these verbs a little bit, and you'll notice that identify is a very low order form. So it should make things relatively easy. All you have to do is be able to identify the form in which something is carried in blood. So we've got a bunch of forms down here, a bunch of substances down here, and we need to figure out what form they come in. Let's get stuck into it. Alright, so this is a centrifuged vial of blood. A centrifuge is a piece of equipment that allows you to separate the contents of a liquid based on density. So we can see here that we've got a large section of blood is just the red blood cells, the erythrocytes, and then we have another large section of it being plasma. We have a small little band here that has white blood cells and platelets in it, and while they are interesting in themselves, they are not hugely what we want to talk about today. We're mainly interested in the stuff that's in plasma and the stuff that's in red blood cells. So let's look at these two today. Alright, so the first substance that we're looking at is carbon dioxide, and there are three forms that carbon dioxide is carried in. The first is actually literally CO2, and it is dissolved inside the water molecules here. So the black molecule is the carbon, the oxygen is the red. So you've got water, which is the hydrogen is the little grey molecules, and then literally just dissolved in it. They you can get that as kind of like dissolved oxygen. You can have dissolved gases. So form 1 is just dissolved in blood plasma. Form 2 is probably one of my favourite biological terms. It's carbaminohemoglobin. It's a great word to say. So we know that hemoglobin can, you generally will associate, you would have heard of the term hemoglobin before. This is the American spelling of it, by the way. There's generally an A over here. So that is, it's a big protein in our red blood cells, and it carries oxygen. But it can also carry carbon dioxide. It can carry carbon dioxide away from muscle tissue where it's being produced, and it can deliver oxygen. So carbaminohemoglobin is 23% of carbon dioxide can be carried when it's attached to a hemoglobin molecule. The majority of carbon dioxide, though, is carried in hydrogen carbonate ions, and this is a hydrogen carbonate ion over here, HCO3-, and it's produced by carbonic anhydrase, and so it's a reaction with water. So the cells produce the CO2. The CO2 interacts with the plasma, and can actually react with the water inside of a red blood cell, and you get this kind of interesting little to and fro reaction where you create carbonic acid, and that can dissociate into hydrogen carbonate ions and a free hydrogen ion over here. So it's this form, HCO3-, which is what this name is here, hydrogen carbonate ion. 70% of that of carbon dioxide is carried in that form. So that's a lot. So that's it's an interesting. That's going to be interesting for later on. So we've got 3 dissolved carbaminohemoglobin, and then hydrogen carbonate ions. Alright, so let's look at this carbonic anhydrase. This is an interesting formula, of which we'll raise it today because it has repercussions for a few dot points later on in the future, and you might look at this and go, oh my god, it's chemistry, but we can't avoid it sometimes in biology. These things are what they are. So we have CO2 plus water can give or yield carbonic acid, and that's this form here, and then that will generally dissociate into hydrogen carbonate ions, and there should be a little H plus in there, and I apologize for not adding that in. That is sloppy on my behalf. But those can reassociate, form carbonic acid, and then that can also reform to another reaction can occur to form carbon dioxide and water molecules. So it's a bit interesting, but it kind of makes sense in the sense that CO2 is produced by our muscle tissue, or by any organ that undergoes respiration, and it goes through these forms and then back to here, and we breathe this stuff out. We breathe carbon dioxide out. So you can kind of understand that there's some chemistry in the blood, and then it goes back out this way. So it's a kind of it's a bit of a journey as we anyway, that's all carbonic anhydrase is, to show you the importance of it. Alright, so we get to oxygen, and another really important substance in our blood, and this is carried in oxyhemoglobin. So this is a hemoglobin molecule, you saw it in the previous picture, and there are 4 sites on a hemoglobin molecule for oxygen to bond to. There's another dot point about hemoglobin, why it's super important, and basically this diagram sums it up right here. Hemoglobin can carry 4 molecules of oxygen. That's pretty important. Alright, rather than just one molecule, it can carry 4 of the bloody things. So that's pretty cool. Oxyhemoglobin is the form in which oxygen is carried. Alright, let's have a look at this. So if you notice what this is, again, it's a centrifuged vial of blood. We now have to talk about water, ions, nutrients, lipids, and nitrogenous waste, and I will lay them out a little bit so they're easy to understand. But it's basically, most of these things can be found in the plasma. So that's this is the previous stuff. The carbon dioxide and oxygen was found in the red blood cells, and the plasma generally contains the water and the other solutes. So our solutes are ions, nutrients, waste products, some gases, and regulatory substances like hormones. Alright, let's have a look. So, I couldn't figure out a way to kind of show you what this is, so I basically just described it like this. So, nitrogenous wastes are basically the product of metabolism, such as respiration, and they're really toxic. We don't like them at all, so we want to get rid of them, and this is essentially what urine is made up of. It's what makes urine smell. You can get a bunch of different forms of them, but the big one that occurs in mammals in our body is urea. So that's the one that you have to remember. But you also can't discount that we also have small amounts of uric acid, small amounts of ammonia, and small amounts of creatine. So urea is the big one. Make sure you've got that one down pat. And then also, if you kind of don't get thrown by any suggestion that there are other forms, because there are, but they're in such minute amounts. Alright, ions. Alright, so some of them can just be like dissolved, like sugar in water or salt in water. They just disappear and not disappear. They are just dissolved into a watery liquid, and plasma is mostly water. So these guys, we have sodium, potassium, magnesium, chloride ions, and sulfate ions can all be dissolved, which is pretty cool. But then sometimes you get ions that can't be dissolved, like irons. Iron basically it oxidizes too easily, and we don't like that chemical reaction in the body too much. So it bonds with a protein called a transferrin, and just a cool little kind of vocab thing here. If ever you see something that's like ferrin, ferrous, ferric, that type of stuff, ferrin is basically the etymology of that word. It means iron. It's the Latin form of iron. So this part, ferrin, of that word means iron. So trans, ion, transporting ion. Yay. Alright, so lipids. Lipids are things like fats, waxes, and steroids. They're also another word that you might have heard in terms of kind of like foods, like people talk about like good foods and bad foods, like good fats and bad fats. So you've got triglycerides and cholesterol. Now, those by themselves aren't bad molecules. It's basically, how much of them do we have that is bad. So basically, what I need you guys to know is that lipids are generally they're insoluble, so they need to be bound to a protein, and they are a lipoprotein. Now, this diagram over here is not meant to confuse you. It's just meant to show you that there are a bunch of different types of lipoproteins, and depending on the amount of lipid to protein, if you've got more lipid, you have these big molecules over here called microns, and they are not particularly good. And the more dense you get, the more proteins, the denser the molecule is, you get HDLs, and they're generally considered the better ones. The reason why these ones are considered bad is generally that they can stick to the walls of our arteries and form like a plaque, narrowing our arteries and causing things like heart attacks. So they're not particularly good. Right, but basically lipoproteins, lipids. Alright, so vitamins and other digestive products are usually dissolved, but like similar to our lipids, some can be suspended in plasma, and then some might require a transport protein too. So you've got all your forms right there. We have carbon dioxide, we have oxygen, we have water, salts, lipids, nitrogenous wastes, and vitamins and other digestive products. So those are the forms. I suggest, you don't have to follow my lead, but if I suggest putting these in some sort of table, maybe with two columns, one called substance and one called form in which it is carried. And look, there's no dancing around the issue. It's a low-level verb, but it does require you guys just to know those forms. So yeah, we'll have a play with that stuff tomorrow in lab. Alright, see you guys.